So this is the study guide review. Now, as usual, for these ones, primary, opportunistic, resident, transient, definition, I don't think anyone had any problem, like match the definition. You will also get plenty of questions about things like, I give you a description of a pathogen. Does that make sense? Remember, I gave you like something causes infection in the hospital settings and can be transmitted, such and such. So you need to tell me, is that opportunistic, is that resident? Is that directly transmitted, indirectly transmitted? Am I clear? Do you understand what I'm trying to tell you? So I give you an actual description of an actual pathogen, okay, with some actual data. And based on what I show you, you have to determine if it's, I don't know, zoonotic, vector-borne, something like that. Are we clear? And please, when you're going to do it during the exam, if you have any questions about like the data or you're not clearly understanding what I'm asking, please ask me about it. I will explain better. I will try my best to clarify the situation for you. Okay? This is pretty straightforward. Which ones have microbiome, which ones don't. Yeah, sterile sites, remember, sterile sites are always more prone to infection. Now, these questions. Taxonomical composition, different body sites, different microbiomes. Same body site, different people, very similar. Not identical, but similar. Okay? Taxonomical composition in health and disease changes. Immune system development, so gut microbiome shapes the immune system, just know it. And gut microbiome shapes the metabolic requirements, metabolic profile of the host. Does that make sense? Just general statements, basically. Can microbial composition one body site affect the physiology of other? Yes. Yeah, I already asked this. For some reason I have the same question twice. Autism spectrum disorder. Just know that, gut microbiome in patients with autism spectrum disorder is changed compared to normotypic patients. Infection means replication. Okay? Now here, okay, some reason, so asymptomatic, unapparent. No clinical manifestation. Communicable, non-communicable. You have to be able to identify if the infection that I exemplify is communicable or not. Nosocomial, iatrogenic, nosocomial hospital acquired, iatrogenic result of the medical procedure. Are you comfortable with the distinction between these two? Because I'm going to ask you about this. No? Yes. So, nosocomial is the infection that is acquired because you're just being in the hospital. So, let's say you visit your friend in the hospital. You're absolutely otherwise healthy. You just visit hospital, but you acquire some kind of infection in the hospital. That was nosocomial transmission. Am I clear? Now, iatrogenic is the result of medical procedure. So if you go to visit your friend in the hospital, you cannot get iatrogenic infection. Nobody does any medical procedures on you. Does that make sense? Good. If somebody, I don't know, receives a prosthetic, and that prosthetic is not clean, and the person develops the wound infection as the result, that's iatrogenic. Okay? Zoonotic from animals. Now, contagiousness, I'm more concerned about basic reproduction number. One thing that I said a few times, and I will repeat it again. Basic reproduction number refers to contagiousness, not the severity of the disease. Does that make sense? Not, I don't know, the speed at which microorganism replicates, just how transmissible the infection is. Am I clear? That's it. It doesn't have to be super deadly, just how transmissible it is. Pathogenicity, qualitative, yes, no. Virulence can be gradual, more or less virulent. These ones. So, host virulence factors versus microorganism virulence factors. Be able to identify which one is which. So if I tell you, exoenzymes, I'm referring to the pathogen. Got it? Median infectious, median lethal doses. Look. You know the definition? Now, about median infectious and median lethal dose, what do you need to know? You need to understand that unrelated pathogens, you cannot really compare their virulence based on, say, median lethal dose, because they're too different. But if pathogens are related, say, different strains of the same bacterium, you can compare the virulence using median lethal dose. Is that clear? You also need to know about this, that higher infectious dose correlates with higher or lower virulence. Higher infectious dose correlates with lower virulence, and vice versa. Very good. Portals of entry and exit. Just understand what they are. If I tell you that the infection is transmitted by a fecal-oral route, what's the portal of entry? Mouth. Exit. Your butt. Yep, that's it. Does that make sense?
I'm sorry, this question is just going to be kind of dependent on how you ask it.
In respiratory transmission, what's the portal of entry? Respiratory tract. No, seriously, it's like, don't overthink those questions. They're very, very straightforward, just to check that you understand what portal of entry is. It's the body side through which microorganism gets into. Like in needle stick, what's the portal of entry? Blood. Not respiratory tract, not rectum, not, urinary tract. Blood. Am I clear? Now, what may happen if microorganism uses portal of entry that is different? Different disease presentation, usually. Acronym TORCH. You don't need to know what it stands for. You need to know that it refers to vertically transmitted diseases from mother to child. Okay? Adhesion happens. First, there are 5 mechanisms for adhesion that we mentioned. Like fimbriae, pili, adhesins, capsules, lime layer. Know them. Viral mechanisms, interaction with the receptor. Okay? Infection tropism, the body side where when microorganism replicates, like neurotropic, cardiotropic. Okay? Receptor for the virus, if certain tissue or organ has the receptor for the virus, that's the organ that virus will infect. Virus cannot infect something without the receptor. Does that make sense to you? Invasion, getting into the cell or tissue. Endosome is often used by virus to invade the cell. Okay. Now, virulence factors. So, exoenzymes, when they are released, they destroy the tissues. 4 classes of exoenzymes that you need to know. Glycohydrolase, protease, nuclease, phospholipase. Now, when I say you need to know, this class of exoenzymes destroys the proteins in the host tissues. That's going to be protease. Does that make sense? You understand what kind of question I'm going to ask about it? Okay? Cytokines storm abnormally high production of inflammatory cytokines. Toxigenicity, toxinosis, toxemia, intoxication. Any questions about the definitions to this 4? That should be pretty straightforward. Okay. differences between exotoxins and endotoxins. So you need to know that endotoxins are produced only by gram-negatives. And then remember the comparative table that I gave you, exotoxins versus endotoxins? Do you remember that? So you have to be able to answer the question, for instance, like which of the following statements is true? Like endotoxins induce antibodies. That's false. Exotoxins induce antibodies. That's true. Am I clear? Three main classes of exotoxins that you must know. Membrane disrupting superantigens and intracellular targeting. We got it? When I say what are the mechanisms of action, well, basically only for one you need to know that actually what's going on. Like, I can give you a description of a toxin. Let me explain. Diphtheria toxin inhibits protein synthesis in the infected cell. What kind of toxin is that? Superantigen, membrane disrupting, or intracellular targeting? Intracellular targeting. Am I clear? So that kind of question. So there are more. So if you can write down the differences between endotoxin and exotoxin in terms of like structures, stability in the environment, whether or not the immune system produces antibodies, that's absolutely more than enough. Good? LAL test is designed to detect endotoxins. It's important because endotoxin is not destroyed by autoclaving and may cause pyrogenicity in a patient. Now, this one, that's really fancy sounding wording here. What you need to know about the LAL test. You basically need to know that when you mix the detection reagent with endotoxin, the more endotoxin you have, the higher the activation of the protease. The higher the activation of the protease, the more color will be produced. That's it. Now, next, following questions about the avoidance of the immune response. What you need to understand. What does capsule do? Prevents phagocytosis. Make sense? Is that understood? Capsule prevents phagocytosis. Proteases break down antibodies. Intracellular infection allows microorganisms to hide from immune response. Molecular mimicry. Microorganism is not recognized by the immune system because it looks like our own tissue. Complement inhibition. Well, it's kind of duh. If complement is inhibited, there's, it inhibits the component of immune response. Leukocidins, what do they do? So you're going to get those questions like, what do leukocidins do? They kill white blood cells. What does capsule do? Prevents phagocytosis. Am I clear? Very basic. The red queen paradox. Microorganisms and humans co-evolve, and usually the infections that have been infecting humans for the longest period of time are least pathogenic, least virulent. Does that make sense? I can. So there are microorganisms that have been infecting humans for millions of years. And that coevolution led to the fact that those microorganisms do not kill people, but replicate very successfully in us. On the other hand, microorganisms that commonly do not infect humans, I don't know, like Lassa virus, that causes hemorrhagic fever. When they do infect humans, the infection is usually devastating because there was no co-adaptation. You see what I'm saying? There was no co-adaptation, so it's all a surprise for an immune system. Make sense? Better? Did I answer your question? Good. Signs and symptoms. You're going to get a list. You have to pick sign or symptom. 5 main stages. Be able to recognize the incubation, the prodromal, the disease, the decline, the convalescent stage. Okay? You have to be able, I give you a description of what's going on, you tell me which stage that is. Difference between localized, focal, and systemic. I'm here. So that's, again, description term. Good? There's going to be a lot in that part. There's going to be a lot description term. Description term. Primary, secondary, all the same. Now, latent, acute, and chronic. And okay, so there's 4 types. Do you understand what's going on? So, in acute, microorganism actively replicates, causes severe disease. Okay, that is usually short term. In chronic, disease is long term, microorganism replicates, causes the disease. In latent, microorganism does not replicate, no disease, it just sits dormant. In persistent, microorganism replicates but causes no clinical disease. Are we clear? Sequelae, consequences. Okay, so we're getting into the transmission story. So what you need to understand, I will give you several descriptions of the disease, actual disease, or actual disease transmission. Do you follow me right now? Based on the information provided, you have to be able to tell me, answer the question like, what is the reservoir here? What is the host here? Okay. What is the route of transmission? Are there any vectors involved? Say, I give you a description of Rocky Mountain spotted fever, and I tell you that it's a disease that is transmitted to humans by ticks. Okay? So you're going to have a lot of answers. Say, one answer will say nosocomial. You think, well, it's transmitted by ticks. Definitely not. Iatrogenic and, zoonotic. Okay, or vector-borne. And like, what is tick here? Is that a mechanical vector? Is that a biological vector? Does that make sense? So you will get those questions and you will have to pick the statement where all words make sense, all qualities make sense. Do you understand what I'm saying? So if I give you a description, I don't know, of urinary tract infection that someone acquired in the hospital, and it will say iatrogenic iatrogenic indirectly transmitted. You think, okay, fomites indirectly transmitted, but is that iatrogenic? No, because it's hospital acquired, so therefore it has to be nosocomial. Does that make sense? All words have to make sense. And for the questions like this, sometimes, when I write the question, I think, oh yeah, they should know this. If you don't understand some aspects of the question, please come to me. I will clarify them for you, absolutely, 100%. Okay? Now, know different types of carriers. Know what fomite and vehicle. Okay? Now, direct transmission routes. Contact, respiratory, vertical, vector. Indirect transmission routes. Fomites, different vehicles like fecal oral, and we got air separated it. Technically, in this case, air is kind of a vehicle, kind of. Okay. But again, if I ask you like which of the following transmission routes is indirect, I give you an explanation with an example. 4 steps. So, Koch's postulates. Just know them. Okay? I can ask you, which of the following is this step, not this step, stuff like that. For this, you will get a question which will basically state the following. You have a hypothetical population of people. So far, does that make sense? Hypothetical population of people. And in that population of people, you have to calculate what's morbidity, mortality, prevalence, incidence, based on the data that I give you. So I tell you, okay, we have 1,000 people. 500 got infected last year. What was incidence? 50%. Am I clear? Out of those 500, 100 died. What's the mortality? Think, okay, 500 got infected, 100 died. That's 100 out of 20%. Clear? Geographical distribution. That is likely be a component of that question about the actual disease. Okay, so I can give you like a distribution, how cases are distributed in the United States or in the world. Does that make sense? And you have to tell if that's endemic, or if that's epidemic, or if that's pandemic or sporadic. Am I clear? Chemical, mechanical barriers. Know them, I guess. Know what they are. Now for innate immunity. You see this? What kind of question can you expect? Which of the following cells is phagocytic granulocyte? You think, okay, phagocytic, we have two phagocytes, neutrophils, macrophages. Which one of them is granulocyte? Neutrophil. Does that make sense? Like the main function of eosinophil is protection against helminths and protozoa. We got it? Know the mechanisms too. So if you see something about, say, basophil and it says phagocytosis, basophils do not do phagocytosis. Am I clear? So again, make sure that the entire statement makes sense. Okay? So this one, cells release in response to injury, leukocytosis, adhesion molecules. So we had a long conversation about the steps of inflammatory response, leukocytosis, attachment, rolling, adhesion, diapedesis, getting into the tissue. Remember that conversation? So first of all, know what stimulates leukocytosis, cytokines and chemokines. Know what stimulates the adhesion of the white blood cells to the endothelium, adhesion molecules. We already figured out that white blood cells follow the trail of chemokines and cytokines to find the place of injury. Does that make sense? So that kind of stuff you can expect. Now, something here. Okay, differentiation of monocytes to macrophages triggered by inflammation. Two white blood cells that play a major role in inflammatory response are monocytes or macrophages and neutrophils. Now, this too, plasma leakage washes away the debris, clotting factors, trap the pathogens at the site of injury. Okay. These two. I will ask you about these ones. Do you understand better now where are pathogen-associated molecular patterns? Where can they be on the pathogen? It's a virus, protozoan, bacteria, doesn't matter on the pathogen. pattern recognition receptors are where? Where are they? Pattern recognition receptor. Which cells? Pattern recognition receptors. Which basically any host cell, including macrophage, yes. Any host cell. So you have to keep it in mind that pathogen-associated molecular patterns are on bacteria, protozoa, elements, whatever, and pattern recognition receptors are on the host cells. Okay, so toll-like receptors, they belong to PRRs. Pattern recognition receptors can be found on the membranes of the cell. Usual effects of the interaction between pump and PRR. Activation of intracellular signaling. That's as far as it goes. Okay, no antibodies are produced. Nothing like that. Just, activation of intracellular signaling. Phagocytosis. That's the question that got stirred you up a little bit. So PRRs on the macrophage surface recognize pumps on the pathogen surface. Okay, that's how phagocyte recognizes what to eat and eats it. So that's pseudopodia. Phagosome is formed and fuses with lysosome, forming phagolysosome. Chemicals that are responsible for killing the pathogen. Hydrogen peroxide, bleach, basically reactive oxygen species, and various enzymes that destroy nucleic acids and proteins and lipids and whatnot. So far, are we clear? Two types of phagocytic cell. We mentioned neutrophils and macrophages. Neutrophils die. Now here, it's a plasma protein, plasma proteins found in plasma, not associated with any cells. You need to know what activates three pathways. They all converge on C3b, and there are several outcomes. Opsonization, membrane attack complex, and increase of inflammatory response. Interferons, you need to know that there are two types, 1 and 2, and interferons are produced by infected cells and can act on the same cell in autocrine manner, adjacent cell in paracrine manner, and distant cell in the endocrine manner. And that interferons interact with the receptor. We clear? We do use interferons for treatment of infections, but limitations are side effects. Effects of interferon on the target cell. Again, it triggers antiviral state or antimicrobial state. It doesn't kill anything, doesn't lyse anything, it doesn't lead to antibody production. Just triggers the antimicrobial state. Are we clear? Now, adaptive immunity. Be able to address the comparison between adaptive and innate immunity. So if I ask you which response is faster, tell me, well, innate is faster, adaptive is more effective, more systemic, but requires an exposure to a pathogen. Does that make sense so far? Good? Humorans, humoral and cellular. Now, for this too, humoral and cellular, first of all, know that humoral is B-cell mediated response and cellular is T-cell mediated. Are we clear about that? Second thing that I want you to know about that stuff. The whole process of generation and maturation of cells. You understand what I just said? B cells, where are they produced? Come on. Bone marrow. T cells, where do they mature? Thymus. B cells, where do they go after they mature? No. When B cells finally mature, where do they migrate? Lymphoid organs. Okay? What do T cells do in lymphoid organs? Why are they in the lymphoid organs? What happens there? what T cells encounter in lymphoid organs. Yeah, but what should happen before they produce effector cells? What should they encounter? What should they meet? Yeah, they should recognize a pathogen. Does that make sense? Think about it, like they generated in a bone marrow, and then they enter 2 police academies. One police academy for B cells in the bone marrow, police academy for T cells is in the thymus. Does that make sense? And then they go policing on the streets, and those streets are lymphoid organs. We clear? Okay? So when I ask about general mechanisms, kind of for review here, so like which of the following cells are a component of humoral response? Helper cells, killer cells, or plasma cells? So humoral, antibodies, plasma cells. Does that make sense? Now, with antigen, know very clearly. Well, you got a good grasp on what can and what cannot be an antigen. Okay. Know that antigenic determinant here is on the antigen, and antigenic determinant is recognized by antibody, and not by like the whole antibody, but recognized by Fab fragment of an antibody. Does that make sense? That's what is drawn right there. We're good? And here's something, again, somewhat uplifting. If you think you are the only ones that cannot tell grasp what recognizes what, it's a problem. And I face it every time we go through this. And this is why I try to give you so many times that same material over and over again. Fab fragment of the antibody recognizes antigenic determinant of the antigen. Make sense? Now, haptene, just small molecule that can be attached to a large carrier. Now, antibody chemically. So, 2 light to heavy chains. Fab region recognizes antigenic determinant. It's variable. Fc fragment binds to the immune cells. It's constant. B cell receptor is technically an antibody. It recognizes the antigen. B cells that carry the same receptor, B cell clone, they all can recognize the same antigen. Negative selection, elimination of the self-reactive B cells. Clonal selection, B cell receptor binds to the antigen, activates the B cell. Clonal expansion, B cell differentiates and proliferates, producing a lot of plasma cells. Activation of B cell by the antigen itself is called T cell independent B cell activation. Sometimes T cells are required for the successful B cell activation, and we call it T cell dependent. Okay. A mechanism of VDJ recombination. Now that's, honestly, it's not a tough one for you, to be honest. It's a tough one for me. Because it's really hard to ask the question about it in the form of a multiple choice. So what you need to understand about this is that we have a bunch of individual genetic fragments. Does that make sense? They recombine and produce a variety of Fab fragments of antibody. That's as far as it goes. Okay? Does that make sense? Okay, yes. So, I had a video somewhere that actually shows the VDJ recombination. Okay, let me talk about it using these 3 markers. It's all going there, at least in the form of audio. So you have 3 different kinds of small genes. The red, the blue, and the black. Those are kinds. Does that make sense? So you have, 20 red, 20 blue, and 20 black. And they're all different. They're all different. We clear? So you randomly pick one red, you randomly pick one blue, and you randomly pick one black. Okay? So number-wise, let's say you picked 5, 12, and 17. Next time you pick, it's going to be 11, 2, and 3. Different fragments. Same colors, but different fragments. Does that make sense? So this creates an enormous number of possible combinations between these V, D, and J fragments. Okay? This red, blue, and black fragments. Makes sense, right? And each combination of these genetic fragments will produce its own individual B-cell receptor with its own individual antigenic specificity. It will recognize a unique antigen. Did I answer your question, Chloe? Make sense? So basically, like, if you can recognize this, it should be fine. 5 major antibody types. You've got a table. You don't need to know any. So basically, you need to know the structural features like dimer, pentamer, the main function. By main function, I mean IgM, first responders, IgA, secretory, IgE, allergy. And you need to know that only IgG can be transferred via placenta. 2 main cell types, plasma and memory, temporal profile. Now, for this, remember that IgM first, and then IgG, okay, during primary response. Now, secondary response, mainly IgG, much more robust and faster, okay. Temporal antibody profile, basically only IgG. IgM are produced very limited amounts. 4 types of humoral immunity shouldn't be a problem. Active, passive, artificial, natural. The one with vaccines I don't think caused any problems. Main mechanisms, we talked about those things like neutralization, agglutination, precipitation, complement activation. So I kind of break it down here. Okay. Now for for MHC. In a nutshell, what you need to know, where these molecules are produced, like type 1 on all nucleated cells, type 2 only on antigen presenting cells, which also tells you need to know 3 types of antigen presenting cells: macrophages, B cells, and dendritic cells. Am I clear? So if you have a question like today, which cell expresses MHC class 1, you have a list in the all nucleated cells. You say, well, all of them, right? Does that make sense? Not only APCs, but all of them. And basically, when I say function, I refer mostly to, like, what is the function of MHC molecules? To present antigen. That's it. Now, for T cells, know the process of activation. Know that T cells cannot be activated by the antigen itself. Okay? Am I clear? So you need a presentation by, say, dendritic cells. And know the difference between positive and negative selection in case of T cells. So during positive selection, you look for T cells that can actually do stuff. They actually work. In the negative selection, you remove self-reactive T cells. So if positive selection doesn't work, you have a bunch of dysfunctional T cells. If the negative selection doesn't work, you have a bunch of self-reactive T cells that will cause an autoimmune dysfunction. CD4, CD8, know which MHC they bind. Know that MHC restriction is basically the fact that type 1 MHC is recognized by CD8, type 2 MHC is recognized by CD4. for T helpers, know the function. Okay? So, like the question today, which of the following is the function of T helper cell or is not a function of T helper cells? Does that make sense? And what they differentiate into. By differentiation, you don't need to know Th1, Th2, just know like CD4 cells can differentiate into regulatory or helper. Okay? And these ones, like cellular and humoral T cells, stimulate the activation of B cells. That's the link between cellular and humoral. T helper cells activate macrophages. That's the link between adaptive and innate. Okay? Tregs reduce the immune response. Here, autoimmune. Now, for cytotoxic T lymphocytes, what I want you to understand is that when we talk about CTLs, cytotoxic T cells, we talk about adaptive responses. When we talk about NK cells, we talk about the innate responses. Know that they cannot phagocytose, and they cause apoptosis either by the receptor system or enzymes and preference. Vaccines, table, 5 types. Okay. Be able to match the type and description. Are we clear? Okay. Hypersensitivities. 1, 2, 3, 4. What you need to know about those. The question will be match the type of hypersensitivity with the mechanism. So if I tell you that in lupus, immune complexes are deposited in the basement membrane, you think immune complexes type 3. Does that make sense? Like, which type of hypersensitivity is that? Or during TB test, cytotoxic response leads to the inflammation of a bump on the skin. So you think cytotoxic, and the response is delayed. Okay, think, okay, delayed, cytotoxic type 4. Make sense? You're going to get only that type of question. So this breaks down much more. I still strongly advise to kind of write it down, because you may not get the entire description. Like, what happens in allergy, like today, okay? Or what doesn't happen in type 4 hypersensitivity. And you have to find a statement that doesn't match, doesn't get into the mechanism. For blood groups, universal donor, universal recipient, know the mechanism of the rhesus conflict. Am I clear? Like, mother is rhesus positive, mother is rhesus negative, stuff like that, Am I clear? I will ask you about the blood groups. Rhesus factor, okay, we got it. Oh, this question. Are there naturally present antibodies against rhesus factor in rhesus negative people? Are there? Like, if you're rhesus negative, do you have antibodies against rhesus factor? No, unless you were exposed, but no. Those are hypersensitivities. Transplants. Okay, so one term that I want to highlight. Okay, twins do not have to be identical. Remember that. So if I ask you the question and I did not specify twins, come to me and ask, which twins are you talking about? Fraternal or identical? Because remember, those transplants are going to be different. If it's between identical, that's isograft. If it's between fraternal, that's already allograft. Are we clear? Okay. Mechanisms. When I say mechanism of the rejection, all you need to understand is that in rejection, the immune system of the recipient attacks the graft. In graft versus host disease, the immune system of a graft attacks the recipient body. Got it? Now, immunodeficiencies. So, only the ones that are here. For acquired. So, I can ask you about DeGeorge. So, you can have a question, which of the following immunodeficiencies is characterized by the lack of T cells? You have to be able to recognize the DeGeorge syndrome. Am I clear? Or lack of both T and B, SCID. Or lack of functional B cells, A gamma globulinemia. Does that make sense? I can ask you this. We talked about some others like immunoglobulin A deficiency. No. Only the ones that are on the study guide. Am I clear? You also have to be able to tell apart acquired versus congenital, or secondary versus primary. Okay, so acquired like cancer or AIDS versus congenital like DeGeorge or A gamma globulinemia. Does that make sense? Okay, few diseases. So I mean, it's all there. It's tsetse fly, signs and symptoms, multiple organ failure and lassitude. It's Africa, no vaccine treatable. Chagas disease, biological vector, reduvid bug or triatomine kissing bug. Signs and symptoms, heart, colon, and esophagus become grossly enlarged. South America, disease treatable, no vaccine. So a few words about HIV and we'll wrap it up. So RNA enveloped, CD4, CCR5, CXCR4. Do you need to memorize them? Only one molecule. You need to know that CD4 is the receptor. I will not ask you specifically about CCR5. What you need to understand, let's say I tell you, remember some people do not have functional CCR5 molecule, remember that? Can they be infected with type 1 HIV? No, because there's no functional receptor. Does that make sense? Know this. A premature release of viral RNA leads to abortive infection. HIV RNA is reversely transcribed into double-stranded DNA by the reverse transcriptase. Know the names. Double-stranded DNA gets incorporated into the cellular DNA by integrase. That's the provirus. When provirus is not expressed, it's a latent infection. Virion has to mature after proteolytic processing. CD4 cell counts reduced in a course of HIV infection, and patient dies of the secondary diseases. Africa has the highest prevalence. North Africa and Arabic countries lowest. Transmission routes. Know them. We talked about them extensively. Best strategies. Sexual education, curbing drug use. Socioeconomic groups that have highest prevalence of HIV in the US. Poor. Okay? And poverty is associated with race, so African Americans have the highest risk. Which steps of virus life cycle? Now, that I want you to know. Attachment, penetration, reverse transcription, integration, virion maturation. Life expectancy for HIV positive patient on heart is normal, just as for uninfected. Resistance is prevented by combining various antivirals. Major challenge for development of HIV vaccine. Virus mutates really quickly. Any questions?